This video will demonstrate how to perform carbohydrates analysis using phenol-sulfuric acid method. Here is the equipment you will need. First, prepare the sample using ethanol extraction. Add 5 grams of apple puree and 45 ml of 80% ethanol into the stomacher bag. Blend the mixture using the stomacher and then transfer the solution into a 100 ml beaker. Cover the beaker with a wash glass and then place it in a boiling water bath for 15 minutes. Cool the beaker to room temperature after heating. Then transfer the solution quantitatively into a 50 ml volumetric flask. Rinse the beaker and make up the volume with 80% ethanol. Mix the solution and filter around 10 ml of the extract for further analysis. Prepare the standards for the phenol-sulfuric acid method as follows. Prepare a 100 microgram glucose per milliliter solution with DD water, using the 1% glucose solution according to your pre-lab calculation. Prepare 10 ml of 0, 5, 10, 15, 20, and 30 microgram glucose per milliliter dilutions with DD water in test tubes in triplicates. Vortex each tube to mix before preparing the next dilution. Make 2 different dilutions, each of 10 ml, of the filtered apple puree extracts with DD water in triplicates, according to your pre-lab calculation. Vortex to mix well. Pipette 2 ml of each standard or sample into a clean test tube. You should have a total of 24 tubes. In the film hood, add 1 ml of phenol using the pipette and 5 ml of concentrated sulfuric acid with the auto dispenser into each test tube. Make sure the solution is dispensed directly into the liquid, not along the side of the test tube. Vortex each test tube briefly. Place the whole rack of test tubes into a boiling water bath for 10 minutes. After heating, cool the test tubes in an ice bath. Vortex each tube and pipette 200 microliters into the microplate. Record the location of each standard and sample in the microplate using the microplate templates provided on Connect. Try to keep the standards in columns A to C and the samples in D to F for easy data manipulation. Read the plate using the TCAN spectrophotometer at 490 nanometers using the preset program phenol-sulfuric assay at 490 nanometers. Save the results in Excel file with session number and group number on the computer and on your USB for further analysis. Dispose the phenol-sulfuric acid waste into a waste bottle labeled phenol-sulfuric waste in the film hood. Rinse all the used glassware and place in the basin. Rinse the microplate, then discard in the garbage bin.